Good morning. I think we've reached a good threshold of individuals or registrants logged in and ready to go. So I just want to introduce you to the AGS, the Arthropod Genomic Symposium X, kind of a takeoff of TEDx, our virtual symposium spring 2022. This is our second installment after doing the same or analogous set during spring of 2021, and glad everybody can join us this morning. So this is a collaborative effort between the United States Department of Agriculture, USDA, Agricultural Research Service, ARS, the Arthropod Genomics Research Community, which is a group of like-minded researchers within USDA ARS, as well as the I5K, a consortium of researchers, international consortium, committed to the sequencing of 10,000 arthropod genomes. And
Brad, excuse me, the slides are not advancing.
Oh, I'm still on the beginning slide.
Oh, okay, sorry.
Sorry, I'm a little long-winded this morning. And INRAE, the National Agricultural Research Institute in France. So let's see this, the overall arching aspect of the AGSx, we're going to be having 4 different sessions. Obviously, you're in session 1, the insect genomic technologies to improve insect food applications, as well as a honeybee workshop that'll be coming up March 9th. And the arthropod genomics and genome engineering in April, and finally wrapping up in May with applications and genomic tools and technology for arthropods. Again, another great set of talks. Things will be coming onto the website, which we're going to cover in the next slide. More details forthcoming in subsequent sessions. And all sessions are at the exact same time. For the United States, 10 a.m. Central, 11 Eastern, and over in Europe, in Central Time for Europe, 5 p.m. So hopefully everybody can join. The same registration, same Zoom link works for everything. So all this is made possible, and I really want to get the thanks up front for everybody who's kind of worked behind the scenes a lot and given a lot of proof. There's a lot of work in the background. First, Kevin Hackett, our national program leader for USDA ARS, for giving the support and the resources needed to conduct this. Glenn Haynes, who is our master of ceremonies, working Zoom in the background, helping with all the technical details we probably couldn't cover by ourselves. Anna Childers, who designed and put up the website, as well as been sending out the Twitter feeds. Pia, unfortunately, can't be with us today to run the video recordings, but she's going to be coming with us in sessions, the subsequent sessions, and getting those videos posted over to YouTube in collaboration with Anna Childers, and Rob Waterhouse for setting up the Slack channel and kind of helping with all the ins and outs and giving the various registration links that we'll be sharing here very shortly. So those informational resources we have available for the AGSx. First of all, there's a website which went out on the couple of links we sent out on email, but it's posted here, and it's going to be also posted into the chat in case you do not have it, as well as the registration link. Obviously, you are here, so you have found the link, but if you want to share that with any friends or colleagues, please feel free to do so. There's the YouTube channel, which has the postings of videos from last year's AGS sessions, but all these subsequent sessions from this year will also be posted there in the future, and you will get an email telling you when they're uploaded and available. As well as the Slack channel, which I mentioned previously, set up by Rob Waterhouse. The link is there and is also going to be dropped into the chat for you to reference to. And there is a link to join, just in case you are a member of the Slack channel. There's a one-time link that you need to click on and register before you can access that resource. And it's good for 30 days. So if you want to try to sign up later after by the next 20 days, it's going to expire. But we will be reissuing different invitations where you can go and re-register or register for the first time in subsequent sessions. So just a little bit of a cue in on how to use Slack for those who are unfamiliar. We're into the digital age and digital or virtual meetings. So we're trying to get a little bit used to this. And Rob posted a reminder on there to go ahead and do a reply to certain threads as opposed to starting new posts or new threads within the Slack channel. No, Rob is on here. I don't know if he's upgraded a panelist. He wants to chime in any kind of additional details on using Slack, but that's just one of the suggestions. We're going to try to have speakers post different materials, maybe manuscripts, in different items and different links for resources to kind of expand the knowledge you can gain from these sessions, as well as the questions and answers will be posted to the Slack channel. So just a slight review of kind of what kicked it off this AGSx in 2021. We had a fairly large number of participants. I think it's 247 independent participants. So those who joined more than one session were not counted twice. And we have several YouTube views. Whoops, excuse me. We're actually have almost 1,000 views right now of all the videos have been posted from last year. So it's great to see the resources being utilized and watched and people learning and gaining knowledge from these resources. So for this point, I'm going to hand over the introduction to Dr. Brenda Oppert, who is the organizer of our session 1, and she's going to give you an introduction and additional details about this session.
Thanks, Brad. Hi, everyone. We began this workshop at the Plant and Animal Genomics Conference in 2017 to stimulate interest and support for genomic resources for the insect as food industry. This year, we have 2 excellent speakers who will discuss the need to incorporate insects as high-quality protein in food for a growing global human population. The presentations will address the development of a new industry to meet the needs of insect farmers and other stakeholders, and the research that is needed to understand food safety, nutrient needs, and other aspects. Next slide. I'm happy to welcome Kelsy Robinson, who is the co-lead on an ARS Grand Challenges Synergies program to develop insects for animal feed, and Christine Picard, who is a site director for the NSF IUCR Center for Environmental Sustainability through Insect Farming. Next slide. So I'll introduce our first speaker today. That is Dr. Kelsy Robinson, a research biologist for the USDA ARS Poultry Research Unit in Starkville, Mississippi. She obtained her PhD in animal science from Oklahoma State University in 2019, where her dissertation focused on the role of antibiotics and dietary ingredients on intestinal immunity and microbiota composition of broiler chickens. Dr. Robinson joined ARS in 2020 as a research associate with the Poultry Production and Product Safety Research Unit in Fayetteville, Arkansas. And she transferred to her current position this past November. During her time in ARS, she has helped to develop the ARS Grand Challenge Synergies project focused on the development of insect meal as an alternative protein source for animal diets, for which she serves as a lead scientist. Her research focuses on improving disease resistance of poultry through the inclusion of immune-boosting dietary compounds such as insect meal. And while Brad is queuing up the video, I'll remind you to please ask questions in the Q&A, and I'll monitor those questions. And the speakers will also be monitoring them to answer your questions. Kelsy will join us live after the video to answer your questions. And I'll remind you that the webinar is being recorded and will be available through a link posted on the Slack channel.
Good morning. My name is Kelsy Robinson, and I'm excited to share with you how ARS is working to advance insects as feed through a USDA Grand Challenge research approach. Before I get into what a Grand Challenge project is and the work that we're doing, I want to share a little bit of background on why we developed this project and what led us to work in insects as feed. This figure comes from a review paper that was published last year in Frontiers in Sustainable Food Systems, and it shows a great overview of the impact of climate change on the food system. We see the negative environmental outcomes of climate change, including increased storm intensity, increased temperature, droughts, flooding, erratic rainfall, and so on. And all of this has an impact on agriculture. In particular, it increases disease severity, causing increased mortality in livestock animals and losses in crops. These environmental changes, along with the increased disease severity and losses, results in lower yields, which causes a decrease in food supply, and the supply that we have will be of a lower nutritive quality. Left unchecked, all of this results in a massive global food shortage. And so it's imperative that we all begin to take steps to mitigate and adapt to climate change. Within agriculture, this means increasing the sustainability of our production to ensure a safe, reliable, and affordable food supply for future generations. And today we're really going to focus on animal agriculture specifically. Within animal ag, one of the ways that we can increase sustainability is by searching for alternative protein sources. Animal diets depend upon protein sources such as soybean and fish meal being the most traditional. However, these are susceptible to factors that affect their ultimate sustainability and kind of limit their sustainability. This includes the negative effects of climate change that we just mentioned, diminishing availability of water associated with climate change, as well as diminishing land for future production as our global human population rises, and increased competition between humans and livestock for both of these products. Anytime that we look at a plant-based protein source, that's typically going to have a seasonal production, which causes shortages and price fluctuations, and necessitates long-term storage, which is associated with some level of spoilage and feed waste. And of course, when we talk about fish meal, we're concerned about marine overexploitation and the impact of that on marine ecosystems. And so while these protein sources have served us well, there is an opportunity here for increased sustainability through novel sources. And that's where insect meal comes in. Insects have incredible potential to increase the sustainability of animal production. They require less water than traditional protein sources. And as we decrease the water input for a feed ingredient, we decrease the water that is associated, the total water associated with animal production. They can be raised sustainably in a variety of climates and locations. And so, especially when we talk about mass rearing of insects, that's done in converted warehouses and barns and factories, making them ideal for urban agriculture as well as diversification of small farms. It helps to buffer against the effects of climate change and means that we can raise insects year-round in a variety of locations, which helps protect against those storage issues, as well as a lot of the shipping issues that we've seen even with the global pandemic. Insects are an excellent source of nutrients, not only protein, but fats and vitamins and minerals. And they're part of the natural diets of multiple animal species, meaning that they're already well suited for these animals. And then one of the things that we are most interested in within our project is the ability of insects to valorize food waste and to take food and feed ingredients that would typically end up in landfills, contributing to greenhouse gas emissions, and instead convert that waste into a usable product for animal agriculture. And so we believe, and we developed this project because we believe that development of insect production models based on using food waste will result in added value for agricultural products that would be discarded, recycling of lost nutrients, and reduced environmental impact. And we're really looking at a circular economy within this project. We're not the only ones that believe edible insects have incredible potential. That's why we're all here for this webinar and this seminar this spring. And as we see, there have been multiple scientific publications from organizations across the world, and these are just a few of them on the screen. While USDA does not have a formal publication like this just yet, we do have multiple scientists that have been working on edible insects for years, long before we established this project. They've been working kind of on side projects and within individual silos. And we developed this grand challenge to help provide them with additional resources and support to really back their research and expand it into a force within ARS that is working to support this growing edible insect industry. And that's how we got to our project, which is formally titled Debugging a New Mini Livestock Commodity, Developing a Model of Insect Production to Demonstrate Their Value as a Safe Solution for Food Waste and Sustainable Fish and Livestock Production. So what is a Grand Challenge Synergies project? Well, it is a synergistic project designed to channel diverse scientific teams to address complex problems of high national importance that may not be solved within the boundaries of a single discipline. So these are not funding opportunities. They are instead highly collaborative 5-year research plans in which we bring together scientists from across the agency to bring their own unique perspective and expertise to solve a problem that we couldn't solve on our own. And that's really what we're doing within this project. We are taking this base of entomology and insect research, and we are applying diverse fields and experience to accelerate the work that we're doing within ARS. Our team is comprised of 42 ARS scientists from 22 research units and 12 national programs. As you can see from the map, our units are spread out across the United States and as well include one unit from Thessaloniki, Greece. And we definitely fit the qualification of a diverse team. And we're really excited about the work that we're being able to do by bringing us all together. From an area of expertise, our scientists fall under the broad umbrellas of entomology and animal production and crop science. Within entomology, we have scientists working on insect genetics, nutrition, diseases, and rearing. And looking at species such as house crickets, yellow mealworm, black soldier fly, as well as many, many others. I believe in total, we have over 25 insect species that our entomologists work with or have worked with. From an animal production standpoint, we have scientists with expertise in nutrition, food safety, health, management, meat quality, as well as agriculture engineering and more. And our species come from poultry, aquaculture, and small ruminants. And then within crop science, we have scientists working in mycotoxin remediation, and they're assisting with the food waste aspect of our project, as well as soil scientists who are participating in the use of insect frass as an organic fertilizer. So that brings us to our model. Our goal, as you saw in the title of our project, is to develop a model of insect production. And this is a very comprehensive model, as you can see. It includes not only insect production, including diets, looking at insect nutrition and waste diets, agricultural byproducts, the safety of those and their suitability for production, but also rearing technology, hardware, mechanization, and our breeding and genetics, looking at insect biology, genomics, transcriptomics, etc. We're also following that insect meal that's produced into animal diets, its safety for animal diets, suitability, how do we process that to get it into our animal diets, particularly those that are, say, pelleted, and how those insect diets affect animal production and health and the safety of our end animal products for human consumption. We're also looking at byproducts of insect production, so insect oil as a possible biofuel or feed source in itself, chitin extraction for chitosan production, and chitosan being a popular antimicrobial, as well as the use of waste and frass, as I mentioned a second ago, as a plant fertilizer for organic crops. And so you can see why we have 42 ARS scientists involved. This is a lot of work that we're undertaking, but we're excited to develop research in all aspects, of the insect industry so that we can continue long after our 5-year research plan to support insect production. Our research is being accomplished through small working groups focused on specific parts of the model. And each group is comprised of scientists with a diverse set of expertise. And what I mean by that is typically we have our insect entomologists combined with animal scientists or crop scientists or a mixture of all three within a group so that we can bring unique expertise and unique technical skills and various different points of view to each project and each problem that arises within our model. Our initial groups have been formed to expand upon that research that is already being conducted by ARIS scientists. So really kicking off this project within the past year, we have taken our ARIS scientists that have been working in silos, and we're looking at how we can expand that research and really synergistically advance it. We currently have 5 working groups: insect rearing, insect diseases, mycotoxin remediation, food safety, and harvesting of wild insects. And so the rest of the time this morning, I'm going to talk about these groups and more specifically, the scientists and the work that's going on within them. So first, we have Dr. Juan Morales Ramos and Dr. Guadalupe Rojas, who are working in insect biology and nutrition. They're part of the Biological Control of Pests Research Unit in Stoneville, Mississippi, and they have over 12 years experience investigating insects as food and feed. They're currently rearing 5 different insect species that have experience with over 20 that I will not even try to list for you today. They have extensive experience working with industry on insect rearing. They've worked with multiple different members of the insect industry, and their research focuses on insect rearing, including mass rearing, mechanization, and diet development, as well as insect nutrition. And they have a couple of great papers that have been published in recent years on self-selection of dietary ingredients as a way to develop effective diets to help maximize growth potential of various insect species. And as well, they work in population biology and chemical ecology. Dr. Ramos and Dr. Rojas help lead our insect rearing group along with Dr. Sharon Schneider. And the members of that group are Dr. Brenda Opert, Dr. Kristen Duffield, and Dr. Joseph Reinhardt. Speaking of Dr. Opert, who has put this webinar together for us today, she, along with the work that Dr. Ramos and Dr. Rojas are doing, she is pioneering ARS's work to develop genomic resources for edible insects. She's part of the Stored Product Insect and Engineering Research Unit in Manhattan, Kansas, and has over 15 years of insect genetic research experience. Her work focuses on insect gut biochemistry, toxin mode of action, and insect functional genomics. And she's previously sequenced the genomes of house cricket, banded cricket, and the yellow mealworm in collaboration with All Things Bugs, which was funded by DARPA. And I don't have to tell everyone here the importance of these quality genomes. Anytime that we're looking at insect genomic research as we move forward, it all depends on having quality reference genomes to come back to. Her future work focuses on sequencing the genomes of additional insects, as well as some RNA-seq work to identify genetic factors associated with increased growth rate and the effect of microbial pathogens on vigor, and as well as some genetic engineering work to modulate larval size. And you can see that she's doing all of that in collaboration with various university partners. Up is our insect diseases group. And this group is extremely unique because insects, just like all other animals, are susceptible to infection by pathogenic microbes, viruses, bacteria, fungi. And however, very little is known about the diseases that impact mass insect production. And this is important because, as we see with our intensively raised livestock, whenever we're mass rearing animals in confined spaces, whenever a disease, a virus, a bacteria, something enters, can rapidly spread through and cause mass casualties that are an issue not only for supply of feed or food, but also for the farmer as they're working to raise these insects as part of their livelihood. This group is led by Dr. Kristen Duffield within the Crop Bioprotection Unit at the National Center for Agricultural Utilization Research in Peoria, Illinois. And she leads the group that includes Dr. Jose Luis Ramirez, Dr. Palme Jasudasan, and Dr. Brenda Opert. And their work focuses on supporting producers by identifying and characterizing entomopathogens infecting rearing insects, such as crickets, black soldier fly, and mealworm, to develop diagnostic, mitigation, and therapeutic tools. And you can see from this picture the difference in a healthy and a diseased cricket, and the idea that our producers very clearly know when they have disease within their populations, but yet have not had the tools to know what that disease is, what the pathogen is, and how to treat that. And so that's where this group, this working group comes in. There's a paper that was published just last year by members of the insect diseases group looking at cricket iridovirus and Akita domesticus benzovirus within diseased and healthy populations of crickets. And what's really interesting about this work, what they found was not only the presence of disease, but the presence of these viruses in both the diseased and healthy individuals. And so it was very common to see active as well as covert infection and co-infection of these viruses within healthy and diseased populations. And so as we move forward, they are working on understanding these covert infections and how they become active infections and how we can then prevent that active infection. Our next group is unique in that they're not looking at mass rearing of insects, they are looking at harvesting wild insects for feed. And this is led by Dr. Alexandra Cheskopula from the European Biological Control Laboratory in Greece. And so for any of you that have been around agricultural settings, whether that's crops in the field or animal agriculture, you know that they're rich, that they are a rich source for wild insects that serve not only as pests, but also a major vector for human and animal diseases. And so this group is focusing on trapping those insects, and we have developed a trap that harvests wild insects and then removes them from the field and converts them into a feed source for livestock animals, poultry, aquaculture, etc. Their work is currently looking at the nutritional and safety profile of wild-caught insects, as well as some feeding studies in poultry. And you can see a pilot study here in the bottom right corner of the screen where they did a two-choice feed test between wild-caught mosquitoes and conventional feed. And these chicks very much, as you can see, prefer the mosquitoes over the traditional feed. They're also customizing the mass trap that they have developed for stakeholder applications under different environments. And they're developing insect decontamination strategies with a field-focused approach so that they can optimize their trap to trap and decontaminate insects so that the final product coming out of the trap is ready for inclusion in animal diets. This team is comprised of Dr. Brenda Opert, Dr. Annie Donahue, Dr. Kamala Arce, and Dr. Lee Kronstadt, along with Dr. Cheskopula, I mentioned earlier. This is the trap that they've developed. In this larger picture here, it can collect 100 times the volume of conventional pest traps, such as the mosquito and the housefly traps shown here. And so we're really excited about this work. And this group actually received the 2021 AGSx award for this work and to really advance it moving forward. Our next group and our next scientist, our next group is food safety, and it is built around the work of Dr. Tawny L. Crippen from TC Crippen from the Food and Feed Safety Research Unit in College Station. Our food safety group is unique in that we are interested in food safety from everything from the inclusion of food waste in insect diets and how that affects the safety of that for insects, to the safety of those insects for animal diets, and then the final safety of those animal products for human consumption. And so this group is involved throughout the insect production process. Dr. Crippen's work specifically is looking at foodborne pathogens in animal and insect protein production facilities, as well as management practices affecting pathogen colonization, retention, and dispersal, interkingdom interactions between insects and bacteria, and carry-on decomposition microbiology. And she's actively doing work in all of these areas, along with her team members, Dr. Annie Donahue, Dr. Kamala Arce, Dr. Pami Jasudasan himself. Speaking of Dr. Annie Donahue, who is a lead scientist on this project on the entire Grand Challenge project, along with myself. She is from the Poultry Production and Product Safety Research Unit in Fayetteville, Arkansas. And her research has historically focused on pre and post-harvest control of food pathogens in poultry, as well as novel sources of methionine for organic poultry. And so those together, that's kind of how she got involved, and by extension, I got involved in insects for animal feed. Her group currently has a SAS grant looking at insect meals, or they are part of a SAS grant, I should say, looking at insect meals as alternative feed ingredients for poultry. And this is really looking at the suitability of insect meal for poultry production. So they have completed an analysis already of the true amino acid digestibility and metabolizable energy values of adult cricket meal and mealworm larvae powders. And they formulated diets using both of those powders, and their process conducting a broiler feeding trial to determine the effect of these diets on growth, as well as on populations of pathogens, food safety pathogens that are relevant to humans. And they're doing all of this in collaboration with various university partners. And as we've shown this in multiple slides, the work that we do as part of this project is not possible without our university collaborators. Even though there are 42 of us on this project, we couldn't get this work done without them. Finally, I'll tell you a little bit about my work. I am interested in the value add of insect meal, looking at the things that we can gain from insect meal by inclusion in poultry diets. That's more than just in sustainability, or even really just increasing growth potential. And so specifically, I'm interested in the fact that insects are rich in antimicrobial and immunomodulatory compounds such as chitin, lauric acid, and antimicrobial peptides, which gives insect meal a potential to improve disease resistance and food safety of poultry. And so my work focuses on determining the antibacterial and immunomodulatory capacity or ability of insect meal, and identifying novel antibacterial compounds within insect larvae. And so really trying to understand how insects can improve disease resistance and improve the health of our animals as we move into a no antibiotic era. To wrap up our project, ARS is really committed to improving sustainability, and that's what all of this comes back to. The figure that you see here is commonly associated with sustainability, in that sustainability means finding methods and products that are beneficial for the environment, as well as for people, and are economically stable. And we believe that insects fall into all three of these areas through their ability to decrease environmental impact of animal production, help secure food supply for a growing population, and boost the economy through product development and job creation. As I've mentioned before, we are really excited to be a part of insect agriculture and the insect industry, and excited to support in any way that we can, particularly through this Grand Challenge research project. Really quickly, I need to say a huge thank you to the entire Insect Grand Challenge team, but in particular, Dr. Kim Cook, who is our national program leader facilitator, who has really helped head up this project and supported it since the beginning, as well as our other national program leaders listed here who support our project, and the scientists who are a part of it. So with that, I will take any questions. Thank you.
Okay, thank you. And
My name is
we are going to take a few questions right now. So I'll ask Kelsy to come on. And once she's on, I will get some of these questions ready for you.
Hey, Brenda, I'm on.
Okay, great. I'm gonna stop my video so they can focus on you. And one of the questions that you indicated you would answer live, I'll just start with that one, is, does the trap release CO2 to attract the insects? And that's the the field trap for collecting large quantities of pests in the field.
Yes, so I wanted to answer this one live because my answer actually is I don't know. So that trap was developed by a couple of our scientists really around the same time that we were getting the Grand Challenge started. So I wasn't involved with developing the trap, and I was actually wondering, Brenda, if you know more about how the trap works. I know you're part of that Harvesting Wild Insects group.
Yes, I think that they do use attractants. So I do think things, probably CO2. I'm not sure of the specific attractant.
I know it is being used for both mosquitoes and flies. So there's some work in, say, dairy cattle or feedlot settings using it for flies as well as mosquitoes in rice fields.
Right. Okay, and Brian Rector has a question that you've answered, but I'll let you answer it for the audience as well. It may stimulate some more discussion because it's really an approach to how we do this research, and is your approach to start with a particular insect species that you know how to rear and see what kinds of byproducts they can eat, or identifying high quality or high priority byproducts, such as great surplus, environmentally detrimental, and then choosing an insect species to rear that are already adapted to eating similar foods? So that's a great question.
It is, and we are starting with specific species. So the most common species that are raised by industry are yellow mealworm, black soldier fly, and crickets. And so we're starting with those and looking at byproducts, kind of testing different byproducts that they can eat. So for example, I mentioned mycotoxin remediation. And so some of the work that we're doing is feeding of mycotoxin contaminated grain. And there's already been some work in this area. We already know that, say, black soldier fly and yellow mealworm can feed on mycotoxin contaminated grain. And so we're really using that as a jumping off point, and then moving from there to other waste streams. And there are companies that are already doing this. And so we're just hoping to take what's already being done and expand that to other waste streams and grow it from there.
Okay. And then we have a question from Priyanka Midapelli. She wants you to address what is one of the major challenges that you face in insect feed and production.
Yeah, so I would say the biggest challenge for insect production and for the industry is scale of production at the moment, particularly when we talk about it from my standpoint, looking at use of insect meal in animal feed. The industry currently can't produce the amount of meal that we need to include in animal diets. And that's really, I would say, the biggest limiting factor to inclusion of insect meal into animal diets, and the price that's associated with production at the moment. Along with that, as we look at feeding of insects, and particularly if we're looking at waste streams, food safety is another main concern. Anytime that we're talking about taking food waste and including that into insect diets and then feeding those, there are concerns, there are regulatory concerns and consumer concerns about food waste. And so those, to me, I would say those are two of the biggest challenges and things that we're addressing within insect production.
Okay, and we have talked about this a little bit among the group, but What are the regulatory hurdles that we're going to face when we try to incorporate insects into poultry products?
Yes, and you would think by now, like you said, we've talked about it so many times, you'd think I'd be better versed in the regulatory concerns or the current regulatory status of everything. Insect meals are being approved for, or the regulations behind them are on a species by species basis. And so, for example, black soldier fly is approved for use in animal feed. Yellow mealworm, I believe they're in the process. Crickets are, I think I saw Kristen Duffield on here earlier. So Kristen, if you know what the status of crickets is, if you'll put it in the chat for me. So all of that is coming on a species by species basis, and we're getting that approved through what's called AFCO. There's kind of two methods. You can get it approved as a generally recognized as safe feed ingredient, or you can go through the more stringent AFCO, and I don't remember what AFCO stands for, approval. And so insect meals are kind of in the middle currently as far as approval through FDA for use in animal feed, with black soldier fly being the farthest along. I don't know if that answered your question all the way, Brenda.
Yeah. And a question from Patrick Schaefer: in terms of species selection, is there any consideration for farming species endemic to the region in which they are farmed? Is there a concern for risk associated with insects escaping from farms into environments where they may lack natural predators?
That's a great question, and it is something that is being considered throughout insect production. There's a video that I love looking at cockroach production in China. And what's really cool about this video is that they've dug a moat around their production facility and filled it with fish that eat cockroaches. So that if they escape, they just get eaten by fish. But that is, it is a concern within production as far as keeping these insects in. And it's part of the reason I believe that the species that that industry is focused on, say, yellow mealworm and black soldier fly. And so I don't know if that's a super great answer to your question, but yeah.
Yeah. So I think we're going to go ahead and go on to Christine, but I thank you, Kelsey, for the great talk and for answering questions. They're continuing to come in, so you can monitor them, and whatever doesn't get answered, we'll transfer everything over to the Slack channel so that we can continue there. And so let me go ahead and introduce Christine. She is our next speaker, Dr. Christine Picard at Indiana University, Purdue University at Indianapolis, which I understand is abbreviated IUPUI, and I know why, because that's a mouthful, so that's how I'll refer to it. Christine has a Bachelor of Science from University of New Brunswick, a master's from University of Toronto, and a PhD from West Virginia University. And she did a postdoc at Texas A&M. She has been at IUPUI since 2011, and she's now an associate professor in the Department of Biology. She's also the director of the Forensic and Investigative Sciences program and site director for the NSF IUCR Center for Environmental Sustainability through Insect Farming. Her research areas include understanding development variation in insects that include blowflies and insects that are agriculturally important, using AI in entomology, and the use of blowflies as environmental sensors. And that sounds like very interesting work, and we'll have to have her back to talk about that. But today she's going to tell us more about her research on developing insects for the food and feed industry. And so I think that while Brad is queuing up the video, I think I have things straight now. That you can ask your questions in the Q&A, and then we'll go ahead and Christine is online, so she'll be answering those, and we'll bring her on afterwards so that she can answer your questions live. All right. And you can see that we're missing Pia a lot today because she's usually the one that takes care of all of this mechanics of getting the videos going, but it looks like we've got it ready.
Yeah, just I'll stay unmuted so you can tell me that everything's coming through just fine.
Everybody, I'm happy to be here
That's
to
good.
virtually talk about genetic and genomic selection in insects as food and feed. My name is Christine Picard. I am from Indiana University, Purdue University, Indianapolis, otherwise known as IUPUI. So participating in this symposium specifically dedicated towards insect genomic technologies to improve food applications, I do want to talk about a few different things. So predicted agriculture is expected to fail to meet the demand for protein that is expected with the increase in populations that are expected. Furthermore, as we all know, we are constrained by environmental concerns. There's currently 1 billion people or more who are underfed. They do not have access to protein. And so we just really need a practical and economic path forward that includes sustainability at its core. And as such, the insects provide this nice means path to a path forward. So the current status, insect farming for both feed and food is a long-established field. And with a continued growing demand for protein and a commitment to sustainability, it is not surprising to see that this industry has essentially exploded in a very short time period. In fact, recent market analyses have indicated that the annual growth of 26.5% is expected over the next 5 years, starting from 2020. And the estimated worth of the industry continues to increase. So more and more companies are coming into this space to try to produce the protein necessary for this industry. And so with this explosion in this particular field, then it is expected that there will be some scientific and technological innovations that occur as part of this. And so what I'm going to speak to specifically has to do with some of the genetic and genomic innovations I expect to see in the future. So here's some example of how much money is coming into the industry, and this was just for last year. So a major company got a large amount of funding in order to create a really large insect farm, as well as they have their own breeding facility. Novafeed is another one who's going to be producing a lot of insect protein in the near future here. And additional companies around the space, including Europe and North America, are raising significant funding because of the recognition that this industry has real potential to change the sustainability profile around protein production. So what is it that we need to accomplish? So really it's optimization of the system itself. What we want to do, and what all these companies want to do, is try to find that delicate balance between desirable traits for both economic or commercial applications, which would include things like development time, size, fecundity, the health of the insect, and even waste stream utilization and the ability to be able to use different streams to circularize the economy essentially around this industry, while trying to remove undesirable ones. And I'll talk about these trade-offs in this presentation. But the idea is to try to figure out, okay, how can we make this the most optimal system so that it is economically viable for others to use this. If it costs more in the early days, that's not an unusual expectation for new industries. But in order for it to be really incorporated into the agricultural space, there needs to be some reduction in costs. And the way that you can do that is by optimizing the system. So with insects, there are pros and cons associated with the mass production of such. So they have very large populations, they reproduce quickly, and they produce lots of offspring. So all of these are definitely in the pro category for anything that you want to mass produce. But they're also in the exact same ones are in the con category too. And that is because of all of these large populations, the ability to reproduce quickly and the ability to reproduce lots and lots of offspring means that there's probably a lot of variation present. And variation is both a good thing and a bad thing when it comes to mass production of insects. Some of the issues around insects is that, physiological time. So if we're just focused on sort of the development rate variation within insect species or among insect species, we can look at how they themselves have optimized through millions of years of evolution at this very specific stage that they're in. So one of the important things is insect development is not linear. So there is an interaction between some of the environmental factors and their development rates, to the point where there is an optimal range in temperatures in order to optimize their rate to the best, to their best ability, i.e. be able to develop quickly, yet still maintaining some of the downstream fitness applications that are needed for the insects. And then you, of course, have all of these interactions with environmental variables, again, with some optimized peak for a lot of these insects when it comes to development time. And then further confounding this is just that there is variation across populations or across geographies that may be contributing to these differences. So if you look at this graph down here below, what you'll notice is that one of these strains has taken significantly longer to develop, but has also increased the size. So here now we have these two competing factors when it comes to mass production of insects. You want the insects to grow fast, but you want them to grow big as well, because both of those are economically or commercially attractive. But what ends up happening is that it's really hard. You can't have your cake and eat it too in some respects, because you get these trade-offs that occur during some of this normal selection that happens. And so in this graph here on the right, what you can see is that survival decreases for some of these strains that can grow faster, whereas the strain that took longer to grow and grew bigger had a much better survival rate. So what happens during its immature environment has a big impact on some other traits that are equally important for commercialization. And that has to do with just this general variation that exists with development. So in this very simplified graph here, you can see that the proportion of the insects that develop during a certain time can vary. Where you have across the mean, you do have the majority of those insects out there, but then you've got some that are faster or slower. And as you bring these insects into an artificial environment, say for mass production, then you ultimately end up breeding a population that will have less variation just because of the process of bringing them in. You are excluding some gene flow and happen to be selecting for individuals that may grow within a certain time limit. Ideally, commercially, what you would like to have is something that grows within a shorter time frame. And then, so that leads us to what happens during this initial process of bringing insects in and producing them on a mass scale. And that is, you're subjecting them to an unnatural or human environment. And you are applying selective pressures to their entire life cycle while they're in place, whether it be developing on a very specific type of feed for the insect itself. You will inadvertently select for the insects that do well on that feed source. If you are collecting eggs during a certain time period, you are selecting for females that can lay eggs very quickly and these may not be the best eggs, to put that in quotation marks. And among other things, getting them used to artificial lights, different, either fluctuating or non-fluctuating temperatures. All of these will have an impact on the insect's life cycle. In this particular example that I've shown here is this really elegant study that showed, there's a very specific region on one of the chromosomes in the black soldier fly, which has reduced heterozygosity, which just means that it appears that there's a selective pressure that has been applied to this area of the genome, which means there's something in this area of the genome that is likely important towards the colonization of the black soldier fly. In this particular area, there's something like 150 genes, and it may or may not be one of these genes that is sort of driving down the heterozygosity in this particular region. But what that tells us is, hey, there's something in this region that may be associated with colonization, which means that these may be particular areas in the genome that are interesting for commercialization processes. So for example, if you have a wild sample, if we can call it a wild sample, one that has not been inbred for commercial reasons, it'll have a lot of fitness related beneficial aspects to its genome, because these haven't been gotten rid of through artificial selection when you bring them in for colonization. However, some of these colonization traits, as we might call them, are really important in order to bring something in for colonization, whether it be something as simple as adapting to artificial lights. So those signatures might actually be present in something like this when you do this type of study, where you try to understand what parts of the genome respond to different, whether it be environmental factors or geographically based differences in phenotypes, and try to learn exactly what it is that contributes to that, because that can be useful in the future. If you have a really fecund strain that just does really poorly in the lab environment or an artificial environment, then maybe what you can do is take advantage of the knowledge you've learned from a study like this and incorporate that into the genome so that the background is really, really genetically strong, yet still has some of these desirable characteristics necessary for mass production. This is another example. This is actually what got me involved as a scientist in this particular area. I was trying to understand sort of the genetic basis of a particular phenotype in an insect. And where I got involved was I was interested in development time, so still am after all of these years. But this was a study that really got it to me. Is in Drosophila, our most well-known insect, what this group did was artificially select for size. In this case, it was size that they were interested in terms of their phenotype. So they had really large flies and really small flies, and then they sequenced across the entire genome, and were able to find these variants or differences in the genome that were significant between these two phenotypic groups. And in this particular case, this was a really elegant study, demonstrated this gene that is known to be associated with regulating cell size. So this demonstrated an example of, okay, now we may understand, at least to some extent, very small extent, some portion of the genetic basis for, in this example, size. So something like that is what got me interested in this. And I've continued working along these lines to try to understand what is the genetic basis that contributes to some of the variation that we see. Which brought me to my own study that I did while I was a postdoc and then an assistant professor here at IUPUI, where we wanted to perform artificial selection on another insect, and we were interested in development time. And so the idea is to try to breed your insects, in this case, to have that desirable phenotype. This has been done in agriculture for millennia, right? So find that plant that grows faster, has bigger fruit, and just breed that from there. So this is artificial selection that you can do. In our case, what we did is we bred our flies. We essentially selected for the ones that grew the fastest versus the ones that grew the slowest. We had independent populations for which we did this through, and then through genomic comparisons, we can look for these differences or these signatures in the genome that may be correlated to this particular phenotype. And of course, that's going to be interesting for a variety of factors. In this case, it was for forensics, but it certainly can be applied to something like insects as feed and food. If we can optimize development for some of these insects, and you can think about the commercial value to that being, being able to increase the number of generations in a year, which in overall increases the amount of waste that you can recycle and or the amount of product that you can produce. And so, unfortunately, with a lot of these selection experiments, though, what ends up happening is you do end up with some fitness trade-offs. If an insect grows faster, it sometimes is not getting all of the nutritional requirements it actually needs as an adult, and may have reduced fecundity, for example. So it's unable to produce as many eggs as maybe a counterpart, which did not develop quite as quickly. However, with some of the tools that we have at our disposal, we can pick and choose the things that we might be interested in trying to optimize in the system. And then this particular slide here just shows the example that you're actually trying to find something that is the essential needle in the haystack. It is really difficult to make these associations between a phenotype and a genotype, in part because in some cases, there can be lots of false positives. So it just happens to look like a SNP is associated with a particular phenotype, but it's actually not associated with that phenotype. In order to get around that, you just need really, really large numbers in order to make your comparisons too. In the past, some of these studies could be costly. The cost is starting to come down, so it's becoming much more feasible to be able to do these types of things. And also, because a lot of these traits are polygenic, they are controlled by multiple genes and multiple regulatory pathways present in the genome, is that each individual change that might be observed in a genome by itself does not actually contribute a lot to that phenotype. It is the combination of many interacting together that contribute to these, to the actual phenotype. And so it's difficult to try to put all of that together. So there's been lots of examples of phenotype-based approaches. I really like this example. There was a population of a honeybee found on an island, which was resistant to mite predation. And of course, that's a really big deal right now. And so the idea was to try to understand what was the genetic basis of this resistance to mites in this particular case. And they performed some hybrid process between the resistant and susceptible populations, and ultimately found different regions of the genomes wherein there's about 100 or so genes in these regions that may be contributing to this resistance to mites. And so what you can do with that, eventually what you can do with that is be able to screen your existing populations or use marker-assisted selection to facilitate breeding. So if you can genotype as these different regions for, your male and female, so in this case, the queen, then you can decide ahead of time which individuals might be the best in order to set up for breeding that may provide some resistance to mites in this case. So we might definitely in insects as feed and food, we're interested in being able to do this. But the problem is some of these trade-offs, of course, and these are well reviewed in the literature. And oftentimes, the trade-offs are economic as well. And that's one of the unfortunate parts. So example in the silkworm is, the less productive silkworm tend to have better survival rates, or in this particular case for bees, increased pollen hoarding had smaller body sizes. probably because they were a little bit harder. Ultimately, hybrid bees had produced foraging efficiencies, and so they were not as good as going out there as their non-hybrid counterparts. And then specifically with the mealworm is that increased pupal weights often led to reduced larval survival downstream. So the bigger the insect was, the reduced survival of those insects. And so, what ends up being the play between these different traits is that ultimately more genetic diversity that's present often leads to increased health and resiliency, which is really good because what you want, especially in a mass production facility, you don't want to risk losing your animals. But that ultimately means that there's more variation in traits. So these are good things and bad things. They're good things for the insects themselves, of course, and for the populations, but they're not desirable when it comes to the production. In order to have this new sustainable protein that will be really helpful for the environment. So really, there's two main problems. So you have to discover the underlying genetic mechanisms that contribute to this trait variation. So often this is done using genome-wide association studies. And there are different types of mechanisms. There can be structural mechanisms, so actual change in the nucleic acid sequence that prevents something from binding or changes the amino acid, which overall or slightly changes the structure of the protein, which in turn can change its function. Or it can be functional. It can exist in areas not considered to be coding per se, but is really important for binding different proteins or regulating different pathways. And these are difficult to determine as well, but there are certainly technologies that exist for us to be able to do any of these things. But this is confounded because how can you cross individuals with desirable alleles across many loci? And if you think about some of these traits that they have so many different binding partners and these huge networks that exist. And so how is it that you can understand things across the whole network without disrupting something that's really, really important? Additionally, some genes, so I'm showing an insulin-like receptor gene here present in Drosophila with many interactions. These are physical interactions of that one protein. As well as, on the regulatory side, there's lots of other things that are happening within a genome. I'm showing the gene here for this insulin-like receptor. And in red are these two long coding RNAs that are present within the introns of this gene. And these can have regulatory function as well. So being able to understand how every single individual part of the genome contributes to the phenotype is a massive problem and is going to take a lot of energy in order to understand it. And so what we can do is take advantage of genetic engineering. We can take advantage of it for knowledge so that we can understand the genetic basis of different phenotypes, as well as if one were so inclined for production purposes. I am not going to talk about any of the ethical or regulatory issues or concerns around genetic engineering here. But I want to talk about it from a scientific standpoint. And that has been used for a long time, both for understanding how different parts of the genome function, and as well as in other parts of our world. And so the actual definition of genetic engineering is the direct manipulation of an organism's genes using some sort of biotechnology. They're known as GMOs, genetically modified organisms. First happened in 1973 when a bacteria was modified. Lucky for us and anybody who's diabetic, in 1978, they started being able to produce insulin using this biotechnology, this genetic engineering. And it was first used in sort of the agricultural space in 1994, the flavor saver tomato, which the tomato was engineered to have longer shelf life. But what's really the powerful aspect of genetic engineering is that by specifically modifying one area of the genome, this one at a time bit, it can allow you to determine or establish the role of that area of the genome. That's the really powerful method that's around genetic engineering, so that you can get an understanding of how all of these different partners interact within a cell or within an organism. And really, ultimately, you end up, depending on whether or not you're using genetic engineering, essentially with the same result. So if we look at conventional breeding here, so you may have something that you are breeding. In this case, a lot of this is seen as from the plant world, but you have a strain present in your lab that maybe has some fitness trade-offs because you brought it into the lab. And so there could be a wild relative or a wild counterpart, a wild strain that may potentially have this beneficial fitness gene that you're interested in introducing back into your colony. And through a series of very specific breedings, you can ultimately end up with the background. The genetic background is your existing strain that you're working with, but you've introduced some new gene from the same, I'm assuming, sexually breeding specimen that conveys it some sort of advantage. Now, you can do this using biotechnology in one of two ways. You can introduce an unrelated organism's genes. The most classic example of this is BT corn, where it is from a different species and gets introduced into the genetic background of your strain and conveys some sort of a fitness advantage. Or you can do cisgenesis, in which you can use biotechnology and the knowledge you've learned about that specific species and make changes in your genome, in the genetic background for which you may like to convey, again, some advantage. And that is referred to as cisgenesis. So it is not an outside species, it is its own species, but you are genetically or you are physically manipulating the DNA. I want to make it clear that conventional breeding does physically change the DNA, but does so in what is seen as a more natural process through just breeding. Whereas there's no breeding that's being used to do this when you use biotechnology. Now, most genetic engineering that has been done with respect to insects has applied to pest species. And the idea is to get rid of them, right? What can we do to get rid of these pest species? I like this figure here because it highlights from this particular paper, it highlights these different areas that we're interested in and where some biotechnology, so, in this case, they're calling them RNA-guided gene drives, but anything like CRISPR technology that allows you to genetically modify a very specific area of the genome can be beneficial. Certainly, if you think about it with human health and getting rid of insects or controlling vector-borne diseases, for the environment by controlling invasive species or even helping threatened species, and for agriculture, certainly being able to manage pests, whether they're insects or otherwise, in a sustainable fashion, as well as especially the idea of non-toxic pesticides and herbicides. At the center here is where you can use biotechnology in order to address all of these. And I would venture to say that insects as food will benefit from these biotechnology advances in order to address all of these things, which includes human health, which includes the environment, and is part of agriculture, in the sense of we can, there's the possibility of being able to change the way the insects are grown so that they can address the environmental concerns. so that they can produce the food that is necessary and contribute to human health. Even if certain insects will contain essential amino acids that could be really an important part of a human's diet. And I do believe that a lot of these biotechnologies will be at the center to drive a lot of this, in part because it provides some speed to the overall process as opposed to traditional breeding mechanisms. So you can move a little bit faster, but also so you can gain a really strong understanding, a foundational understanding of what it is within the biological sphere of that species and what these different, how this variation is present and how it is regulated, and what's important in a commercial aspect. So as I conclude a little bit, I want to touch on these three species here. So you've got the mealworm, the black soldier fly, and the cricket. These are sort of known as the big three when it comes to the insects as feed and food, although there are something like 3,000 species that are currently produced either for feed or food purposes. But these are the big three that each have their own advantages and disadvantages. And there are examples where work is being done in order to optimize these systems. I have personally worked on some genetic understandings of variation within the mealworm. And there's been work done on the black soldier fly, and the cricket, I hope, is soon to come as well. And I just wanted to highlight one study that was fairly recent with the black soldier fly genomes. Recently, the genome came out, and what they did is they specifically targeted 2 different genes present in the black soldier fly that may or may not have some sort of a commercial application to it. One gene is related to insulin signaling pathways. So this has to do with regulating growth and regulating molting between stages. And specifically what they noticed when they knocked out this gene in the black soldier fly was that the fly lasted much longer in the pupal stage, and that length of time in between and for each stage meant that it was able to feed more, which made it ultimately a much larger insect in this particular case that you can see from here. So that is simply from modifying or knocking out a single gene in that species, and you get this fairly significant change in phenotype. And this is another example here, which was another gene, vestigial, which is a wing gene. And essentially, they were able to show that the wingless flies, in this case, that had the genetically modified wing gene, were still viable and still produced offspring, which meant them to be a strong candidate to keep in the lab, or to keep for mass production. One good thing about having a wingless fly is that the escape routes for it become less prolific. So if a fly can't fly, then it is harder for it to escape from a facility that is breeding these. And there's cautionary tales about, especially with the black soldier fly, getting out there into the environment, especially a breeding facility that may have selected for some traits that, if out in the environment, can have some ecological consequences. So there's a lot of interest in making sure that individuals that are being mass produced are not being released into the natural environment. And so this is one example where there's a nice commercial application using, in this case, they use CRISPR in order to knock out these genes. So coming back around to this idea of looking at these comparisons and looking at these colonization type genes, I would venture to say this is a really good place for us to start as a community to try to understand what is the genetic basis for the phenotypes we see in some of the lab-based or the mass-produced based strains, both good and bad. And I think that's what we need to understand. If there is reduced fecundity after being brought in for mass production, what is contributing to that in a genome? We should understand that. That'll give us both the tools to be able to genetically screen organisms to set them up for breeding. So you're not doing any genetic modifications, you're simply screening your populations for the ones that have the most desirable phenotypes. You are unlikely to get all of them, but you might be able to get quite a number of them. And a lot of these screens are fairly cheap, and you can do these on a large scale, and there's so many insects that you can do that with. So that's A, or B is being able to maybe knock something out, like that vestigial gene example that I just gave you, which gives you the opportunity to do something that, you know, with these loss of function or these knockout experiments with CRISPR is you're simply, you know, cutting the DNA in a very specific area and then allowing it to rejoin or fix itself, repair itself, but it does so by making some mistakes. And so you're not introducing anything foreign in that particular example. However, you are physically manipulating the DNA by some exogenous source. And so it still falls under a genetic modification. Again, I'm not here to comment on any ethical or regulatory concerns about using genetic modifications. I'm here simply to talk about the science. So I will finish with just a short plug. As this industry continues to grow on the commercial side, academia needs to catch up and certainly address some of the concerns that this growing industry is going through. One of the worst things that could happen to this industry is if something bad happened, if there was a safety a problem, it could certainly derail this industry rather quickly. So through NSF partnerships and funding, we've established a Center for Environmental Sustainability through insect farming, and it's a collaboration right now of 3 institutions. We are open to more institutions joining this. In fact, we welcome more institutions joining this collaboration, and we work directly with industry that's involved with either producing the insects or using insect protein in their own production facilities. And the idea is to shorten the gap between what we do in academia when it gets out to industry so that they can use it and innovate from that. So the foundational research that's necessary to help inform the industry. So we're always open to more industry members that fund the research, as well as more academic partnerships that can really put this center together. So with that, I'll stop here. Thank you very much for your time.
Okay, thank you, Christine. And I'll ask you to go ahead and invite you to come on live, and we'll have a few questions. And while we're waiting for Christine to come on.
I'm here. Sorry, I didn't know you were waiting for me.
Sorry, yeah, because we have some questions that are being asked, and I'm going to go ahead and stop my video and concentrate on these questions. Amanda Stalkey asked a question that you've answered, but I thought it would be good to answer it online because these are great questions. Do characteristics of the insect genomes make them more or less amenable to selective breeding? For example, does the larger size of cricket genomes make it more difficult to predict breeding outcomes?
Yeah, and so my answer to that was I don't fully know the answer, but I'm inclined not necessarily to think that it will. I think with enough knowledge, you should be able to screen any genome and try to make some decisions for breeding. the size of the genome, I don't think has an impact, because certainly there's been, genetic selection happening in many other animals and plants that have much larger genomes than our insects. But then I did write, I think the bigger issue is how little studied or understood insect genomes are relative to the amount of variation there is, or just the sheer number of species. So I think that's what's holding us back more than anything else.
Okay. And I would agree with that. In larger, here, this is a question from an anonymous question. In larger livestock, it is relatively easy to take up samples like blood samples for genetic screening without killing the individuals, which then can be selected for the breeding program. How could you do the same in screening insects without killing them?
Yeah, this is a good question. There are lots of opportunities for that, depending on the species. But for example, in our lab, especially with blowflies, but with any fly, we can get DNA from the pupal casings. Or with mealworm, I've worked with some mealworm work where you can just clip like a tarsus from the adult fly and get enough DNA to be able to screen in that situation. And even there's work being done in museums where they are just taking the insect and basically like, these are pinned insects in this case, but you must be able to do it with live insects, like just sort of washing them a little bit in a liquid and being able to get enough DNA that way. So I think there's tons of opportunities for us to be able to genetically screen the insects without killing them, because then that, you want to be able to screen them and then use them if they're of interest to you.
Okay, thank you. And then Kevin Hackett has a question about the black soldier fly. He says, will there be work towards a pangenome, BSF genome and genome study? And how much variation do you anticipate in the greater BSF world?
Yeah, it's a good question. BSF's an interesting species for sure, because a lot of the I think mass production strains, a lot of them originate from a single strain, so this so-called shepherd strain. And so there was a publication that came out not that long ago, last year, year before, that looked at the population genetic structure of BSF across the world, and there is variation that exists for sure. But I don't think that variation is being tapped into on an industrial scale. But there may be interest to develop that some more for sure. I think a pangenome would be awesome to get.
For any insect, right?
Yeah.
And I had a question as far as in the trade-offs between size and developmental time, which is just very common among these insects that we're studying. What do you think is more important in insect production for food?
Ooh, that's a good question. Yeah, I actually don't know the answer to that. And I wonder, part of me wonders what folks that are producing the insects actually think in this respect. You could probably do some math to figure that out. So if the insect gets a little bit bigger, but takes longer, but you produce a greater unit of protein per, energy input and feed input, then that might be more valuable versus an insect that grows faster, but is a little bit smaller. So I haven't done any of those types of calculations, but I bet somebody out there has. And so if anybody does know that on the insect production scale side, it'd be great if you could put that into the Slack channel.
Okay. All right. I think we're a little bit past 11:30 my time. And so I think we've covered a lot of territory here, and we're going to go ahead and wrap up this session. But I encourage you to continue to post on the Slack channel. We can continue the discussion. and I appreciate everyone's attendance today. I know that we are getting people from all around the world, and so it's really rewarding to have a space like this where we can talk about these issues related to insect genomics. And so I appreciate Kelsy and Christine. It takes a lot of time, and I really am grateful for them putting together great talks and putting themselves out there for you guys to ask more questions. So thank you all, and thanks Brad and Glenn, and I appreciate everyone that's behind the organization of the meeting. I'll remind you of the next session, which is in March, and that is the honeybee session. So I know that Sonia and Alan are putting together a really good program that's going to be posted soon at the I5K website, and we look forward to that workshop as well. So thank you, everyone. I hope you have a nice remainder of the day or night, wherever you are, and appreciate you attending. Thank you.